Here. Welcome everyone to the IMO seminar. It's a pleasure to host Dr. David Hormuth today, who is a research associate at the Odin Institute for Computational Engineering and Sciences at the UT Austin in Texas. He's a member of the Society for Mathematical Biology and the Mathematical Oncology Subgroup Executive Committee as of fairly recently. David and our research is very synergistic, and we are highly interested in the great work he has been doing over the past few years. So he's particularly interested in integrating predictive mathematical models with patient-specific data to help guide cancer care, which I think speaks to a lot of us. He's very interested in image-driven biophysical models of cancer, tumor growth, treatment response, etc., and looking for novel ways to adapt therapy. So he's really interested in predicting how tumors develop forward in time, and then how to choose the right treatment, how to optimize the treatment, and improve outcomes for patients. And I hope that at least a subset of what I just mentioned will be the topic of David's presentation today. So with that, I'm very happy to hand it over to you, David. Thanks for joining us.
Thank you. Yeah, that's a good setup for what I'm going to be talking about today. Let me just get my Zoom set up here. Y'all can see my PowerPoint screen, correct? Okay, great. Yeah, so yeah,
my
name's David Hormuth. I'm here in Austin, Texas, and I'm going to talk to you a little bit about using quantitative magnetic resonance imaging data to kind of enable these predictive models of tumor growth in response to radiation therapy. Sorry about that. So my background is in biomedical engineering, specifically kind of medical imaging and cancer imaging. And I'm a member of the Center for Computational Ecology, which kind of my research effort is kind of spent half of the time doing computational work and the other half of the time doing experimental work. So that means some of the times I'm developing these mathematical models and calibrating them, and then, running the simulations on kind of our high performance computing resources. And then the other part of the time I'm doing small animal preclinical imaging research and kind of designing experiments. Right now, we're not in the lab doing experimental work, so a lot of my experimental energy is being focused on, bread baking at home. So the overall working hypothesis for our group is that there are readily available imaging techniques that can provide the data necessary to initialize, constrain these predictive models of tumor growth and response. The idea is that we'd like to collect images, MRI, PET, CT images before, during, and after treatment, and use these images to calibrate or assign model parameters for individual patients. We can then use these calibrated models to predict treatment outcomes, maybe optimize treatment, or adapt treatment kind of in real time for these patients. And within this kind of overarching thing of trying to define these patient-specific models of tumor growth and response, my main focus is really on modeling response to radiation therapy in high-grade gliomas. So within the Yankee Love Lab, which I'm a member of, there are kind of 3 different areas that we're working on this kind of project. One is at the in vitro scale. So one of the projects I'm involved with is this time-resolved microscopy-driven model of response to radiation therapy. And this is a project that's led by a graduate student in the lab, Junyang Liu. And then at the in vivo preclinical scale, this is one of my main projects that I'm going to talk about today. We're trying to kind of develop the experimental and computational framework to use MRI data to drive these models of response to radiation therapy. And then the most recent thing we're doing is actually translating the stuff that we've developed in the preclinical scale to clinical imaging types. So here's kind of the outline for my talk today. I'm first going to provide just a little background on radiotherapy, an introduction to the imaging and the modeling that we're using. And then I'm going to have a preclinical example, a clinical example. And then if you aren't already eating lunch, you guys can go have lunch after this talk. So first, a little kind of history of radiation therapy. So X-rays were first discovered in 1895, and within a year, People are already beginning to try to use X-rays to, image the body and also to perform radiation therapy and to try to treat lesions. This has been around for over a century now, and this technique has continued to evolve and have a strong foothold in treating of cancers in oncology. So over 50% of patients receive radiotherapy either as a therapeutic or palliative treatment. And with chemotherapy and radiotherapy, it's one of the three main options that are typically presented to patients. It is significantly part of the standard of care for high-grade gliomas. Typically, patients who have high-grade gliomas receive a maximal allowable resection. The goal behind this is to remove as much of the tumor as possible, but this is not always the case. The tumor itself is highly invasive, so there are tumor cells that we might not actually be able to remove surgically. And then also, because of the sensitive nature of where this tumor is located, it is sometimes impossible to actually access different parts of the brain or to remove the whole tumor without damaging functional elements of the brain. So after surgical resection, patients typically receive temozolomide and then radiotherapy to treat the residual disease. So because my focus is really on radiation therapy, I really wanted to give you a little background on how radiation therapy treatment plans are currently made. So a big picture look would be to say radiation plans are based off of these anatomical and structural images. So things like contrast enhanced MRI, T2 weighted MRI, FLAIR MRI, and these images report on the extent of disease. And so from these image types, they can define tumor treatment volumes. They can identify the bulk or clinical disease. So this might be that contrast enhancing lesion. And they can also identify that invasive or subclinical disease. So this is the area where we see some abnormalities in the imaging data, and we're suspecting that there is some invasive tumor spread there. And so from these tumor volumes, they can develop these treatment plans for highly conformal radiation therapy, where basically they're going to pass radiation beams in different directions to try to treat this tumor region while decreasing exposure to non-diseased parts of the brain. A more recent approach is trying to move these treatment plans from just structural or anatomical images to incorporate some element of the tumor biology in the treatment plan itself. So luckily, there's a lot of imaging data that are sensitive to features that are relevant to tumor growth. So one example would be diffusion weighted MRI, which might tell us something about tumor cellularity. Dynamic susceptibility contrast MRI, which provides some estimate of tumor perfusion or blood volume fractions. There are PET imaging techniques such as FLT or FDG PET, which tell us something about tumor proliferation or metabolism. And then F-MISO PET, which tells us something about hypoxia. So these imaging measurements could also be used to take our anatomical design, identified tumor volumes, and then overlay maybe an additional volume of saying, this region right here, this region in black is let me get a laser pointer. This region in black right here is maybe this hypoxic or potentially less responsive region of tissue. And from this, they can develop a more advanced treatment plan, maybe use something like intensity modulated radiation therapy to boost the radiation that is delivered to different parts of the tumor, depending on that biological response to radiation therapy. So we have our structural anatomical plans. We have these biologically guided plans. And the question might be, what is missing in these plans? What are something that we can include, improve on these plans? So for the structural and anatomical based plans, these tell us where the tumor is. For these biologically guided plans, these tell us what areas might be less responsive. But the big question is maybe how do these things change over time? If you make these measurements at the beginning of radiation therapy, these regions are likely to change during the course of therapy as the tumor is either growing or responding to that treatment. So for these anatomical plans, there's a lot of effort working, focused on correcting these treatment plans for deformation in the tissue or maybe respiratory motion. This is not so much an issue in brain, but in other organs, they are correcting for respiratory motion. And then also you could maybe adjust these treatment plans to just account for growth or shrinkage of the tumor beyond what was kind of expected when they developed that initial treatment plan. Then for the biologically guided plans, these, if there were no treatment being applied to these tumors, this area of hypoxia might change or evolve over time, or we might have areas of increased vascularity. And all these things would influence response to radiation therapy. But in the presence of radiation therapy, these things are also going to change. They're either going to shrink or expand in response to radiation therapy. And these are things that could be included in kind of considerations when developing a new treatment plan. So part of my work is trying to move us towards these kind of patient adapted or optimized radiotherapy plans. And I don't know about anyone else, but if you've gone to kind of search for adapted radiation therapy or kind of optimized radiation therapy, it really depends, depending on who you're talking to or what conference you're at, you're going to see a very different response. So I feel like 75% of the abstracts that I've read or posters that I've gone to look at, I've really focused on kind of this just altering the treatment plans to account for the daily shape or change in the tumor. So this is a huge field of medical physics, just making sure that the treatment plan that you've prescribed is what is actually going to be delivered to that patient. So this is very important work, and we need these type of adaptive radiation therapy plans to do anything else that we'd like to do. And then a smaller subset of that is probably these biologically informed or biologically adapted radiation therapy plans. So these are things like adjusting dose to boost radiation therapy to rest responsive regions, maybe adjusting when treatment is actually delivered to kind of improve the outcomes for these patients. So to be able to kind of perform these adaptive radiation therapy plans that are kind of accounting for a patient's unique tumor biology, we are in need of a model of kind of the spatial and temporal response to radiation therapy. And my kind of hypothesis is that if we have this model, spatial temporal model of response to radiation therapy, we could then use this to simulate in silico different treatment plans and adapt the overall radiation therapy plan that is going to be specific for this individual patient's tumor biology. So this is kind of this model and image guided plan that I have that I'm thinking about for our research. So the idea behind this is that we'd have a baseline image. So this is going to define kind of the structural and anatomical plan and the biologically guided plan, like we see above, where they're going to begin treatment and then maybe have a second MRI during the middle of treatment. And with these two images, the baseline and the mid treatment MRI, and we're going to use these two imaging types to calibrate our model. So this is going to tell us something about how well the patient is responding to that radiation therapy after just 1 week of radiation therapy. We could then use this to refine our treatment volumes to kind of identify regions that seem to be less responsive to radiation therapy. And then we can repeat this process while we're delivering radiation therapy to continually optimize and adapt the treatment protocol to kind of best control for that tumor growth. So the overall goal of kind of the techniques I'm going to talk about today is kind of moving towards these this kind of model and image guided optimized radiation therapy. So the focus of my talk is really going to be on integrating these imaging measurements with these mathematical models to make predictions of response.
Hello, David. I have a quick question.
Yeah.
So I'm just wondering that the time scale here, you're talking about while you're giving radiation, so in the order of like an hour?
No, so the time
over days.
Yes, so the idea would be, so with glioblastoma, radiation therapy is given in fractionated manner. So potentially you could take so the radiation therapy is typically given on the weekdays, Monday through Friday. So you could potentially have an MRI on Friday after 1 whole week of radiation therapy to kind of quantify how the tumor is responding to that radiation therapy, use that information with your model, and then when you return to treatment plan on Monday, you can provide an updated treatment plan. That's kind of the time scale that I'm thinking of.
Okay. Okay. So you're looking at between different fractions.
Yes. Yeah. Between
Okay,
different
yeah,
fractions.
got it. Okay, I get it. Thank you.
So to kind of begin to model kind of response to radiation therapy or tumor growth, we kind of need to look at what are the different kind of how radiation therapy interacts with tumor cells. And let's see, there we go. So immediately after tumor cells are irradiated with this lightning bolt of radiation therapy, ionizations occur very quickly within 10 to the minus 15 seconds. DNA damage occurs within a microsecond. And some of these cells receive significant cell death, or receive significant damage that they undergo death or senescence following this treatment. And then some of these cells, actually a vast majority of them, undergo repair process. Under this repair, some of these are successfully repaired and can proliferate. Some of these fail to repair that DNA damage and undergo a kind of late death or die at a later time point. This could be due to apoptosis, maybe mitotic catastrophe. But one thing that's kind of interesting about radiation therapy is that even if they repair correctly and are able to proliferate, they may proliferate another generation and they might die out at a later generation due to that initial radiation damage. Or even further down the line, they might become senescent and unable to proliferate. And so one of the main ways that people kind of assess response to radiation therapy is really this clonogenic survival. Since we aren't really able to see, since there's not a really immediate response to radiation therapy, often in these early timeframes, we're looking at the ability of these tumor cells to form a colony down the line. So this is one of the things that kind of makes modeling response to radiation therapy tricky. So we have a very fast delivery of treatment, but then we have this potentially long delay in response. The next thing we might kind of consider when we're building these models are things like the 5 R's of radiobiology. So these 5 R's consist of repair. So cells within the tumor might have different repair mechanisms or might have kind of stronger repair mechanisms that are more resilient to damage. Different tumors might have different growth rates. So if you have a very slow growing tumor you're treating with radiation therapy, maybe the standard of care approach will work just fine for that tumor, whereas a very fast aggressive tumor might be less sensitive to, or might regrow faster and would need additional treatment. Reoxygenation, which is kind of an area that our lab is definitely looking into, is basically radiotherapy kind of preferentially targeted well-oxygenated, well-vascularized tissue. And with each dose of radiation therapy, it kind of removes or kills off that well-oxygenated tissue. And it allows, it kind of stimulates increased angiogenesis, which then allows kind of reoxygenation of tissues that were previously hypoxic. So now these tissues that have been reoxygenated can now be treated with additional rounds of radiation therapy. And kind of the dynamics of this reoxygenation process would be kind of important to understand in kind of designing an adapted, optimized therapy. Next would be redistribution. So kind of one of the main reasons they give fractionated therapy is to target cells at different points of their cell cycle. So depending on what part of the cell cycle that cell is in, it might be more or less responsive to radiation therapy. And then finally, radiosensitivity. Some regions within the tumor, or maybe even particular cells, just might have an inherent different radiosensitivity to radiotherapy. So some of the models that we're developing at kind of the in vitro and in vivo preclinical scale consist of elements of these things, and we're hoping someday to kind of combine them together. So what kind of things should we include in our models that we're building? So we could consider including all these 5 R's of radiobiology. We could also consider including maybe like the hallmarks of cancer. But one of the big questions we have when we're kind of going into model building is how are we going to assign model parameters for this for individual patients? How are we going to make these models subject specific? So one of the first projects I worked on in graduate school was basically, I got a list of papers and a list of models to look at and trying to kind of combine them all together to kind of get just an intro to, coding up in the finite difference method and implementing these models. So, we developed this model of, tumor growth and angiogenesis and oxygen consumption. And, if we watch the simulation, this, recapitulates a lot of those kind of hallmark characteristics of tumors. We have this high cell density ring, this low cell density core. We have distribution of oxygen. And let's see, I think if I change, And one of the things that's challenging about this type of model is that there are a lot of different things that we need to know or measure to kind of initialize this. So we first need to know the initial conditions. So things like the total number of tumor cells, the blood vessel distribution, the oxygen concentration, maybe an initial map of cell state, so whether or not the cells are proliferative, quiescent, or necrotic. We also need to have model parameters that are describing things like proliferation rates, death rates, diffusion coefficients. And all of these things are really difficult to measure for individual tumors, let alone non-invasively. So we started with this really complex model, early on in my graduate work. And kind of shortly after this, we started looking into how can we actually parameterize these type of models with imaging data. So, we made a huge list of the different type of imaging measurements we can acquire and begin slowly implementing those in our models. So the next couple slides are going to just be kind of a little brief, introduction to some of these imaging measurements that we've looked at, and before we get to the actual modeling work. So there are several different types of imaging measurements that we can get and that our lab has looked at. The main one that our lab works with is diffusion-weighted MRI, DW-MRI. And this is an imaging technique that is sensitive to the diffusion of water molecules in tissue. So if we look at this bottom box, you can imagine a voxel of cells. These little kind of circular things are cells, and in between is the extracellular, extravascular space. And if you perform a diffusion experiment, the water molecules would have very restricted space for it to move. And so if we were to measure the diffusion coefficient of water in this tissue, it would have a restricted or lower apparent diffusion coefficient. As tumor cells die, there is more space for water molecules to move around, maybe even move it in and out of the cells themselves. So this causes an increase in that apparent diffusion coefficient over time. So it's going to begin to shift to the right. And then finally, if we have a voxel where there's a lot of cell death or necrosis, or maybe even no tumor cells at all, this apparent diffusion coefficient is going to approach the diffusion of water in free space. So an example we have here is from breast cancer. In this image, we have kind of fibroglandular tissue, and then we have our tumor tissue. And then when we look at the apparent diffusion coefficient map on top of this image, we see very distinct regions of the ADC. So we have areas that have a low ADC, so maybe indicating high cellularity, and regions that have a high ADC, maybe indicating regions of low cellularity. The next technique that our lab kind of uses a lot in our work is this approach called dynamic contrast enhanced MRI. And in this approach, we inject a contrast agent into the body. The contrast agent moves through the blood vessels. And for the case of brain tumors, this contrast agent remains in the blood vessels in healthy brain because the blood-brain barrier is intact. We get a slight increase in the signal intensity due to the contrast agent just being in the blood vessels there. And then when it hits the tumor, some of that contrast agent remains in the blood vessels. Some of it extravasates from the blood vessels and accumulates into the tissue space. And one of the things that's kind of nice about these experiments is we can analyze the measurements that we get from this experiment using a two-compartment model like this one right here. where we have a compartment for the plasma space, a compartment for the tissue space. We can estimate parameters for K-trans, which is kind of like a perfusion or permeability of the blood vessel, or KEP, which is the movement of the contrast agent from the tissue space back to the plasma space. And then we can also estimate the plasma volume fraction. So let's see. Okay. So here is an example in one of the animals that I've worked with where you can kind of see the rapid increase in contrast agent and accumulation of the contrast agent within the tumor. So with these experiments, with DC MRI experiments, we get a measurement at each voxel within the brain. And so for the kind of purpose of looking at what a kind of typical curves look like, I place these regions of interest over kind of different tissues, and we're going to look at what the kind of characteristic curves for these different regions look like. So this blue ROI corresponds to the muscle region of interest. So if we look over here on our signal intensity plot, we have about 5 minutes of images collected before we inject a contrast agent, and then a rapid uptake in the muscle, and then it kind of slowly washes out. The red region here is healthy appearing brain. So once again, 5 minutes of kind of no uptake, and then we get a slight increase in signal intensity. It's very, very small, but because the contrast agent is not leaking out and kind of accumulating with in the tissue. But since there is quite a lot of blood vessels in the brain, we do have a increase in signal there. The yellow region is part of the tumor here, and so we see a rapid uptake in signal intensity. And then this white ROI corresponds to this purple line over here, where we actually see kind of a slowly increasing signal intensity over time. So this is a really good example of a part of the tumor that is not vascularized. And actually, the contrast agent is probably diffusing from the kind of well-vascularized core into this kind of potentially necrotic region.
David, can I ask before you go on,
Sure.
What's the resolution of a voxel here? Like, how big is it?
Yeah, so this entire image is 32 by 32 millimeters. So, and each pixel or each voxel in this image is 0.25 by 0.25 millimeters, and then a 1 millimeter slice thickness.
Okay, so that's obviously a lot finer than you get in the human setting, right?
Yeah, so I mean, probably the highest resolution that we typically get for maybe our breast studies are maybe 0.5 millimeter isotropic resolution. But you know, with those, as you increase the resolution, it's going to affect kind of the signal to noise ratio of those images, or the quality of the images.
Sure. Thanks.
No problem. Another imaging technique we could use are things like MR elastography. So in these type of experiments, they can kind of apply some kind of force onto these tumors, remove that force, and they perform kind of estimate the tissue elasticity, or maybe the shear modulus or Young's modulus of this tissues. And this could be used to either assign mechanical properties for the tissue or for the tumor, or maybe look at differences in stiffness between lesions. And for radiation therapy, one of the approaches that we're kind of really interested in is kind of tissue oxygenation. So there are PET approaches to measure this tissue oxygenation or tumor hypoxia, such as 18F-meso. And then there's also MRI approaches that are kind of gaining traction right now, such as tissue oxygen level dependent MRI or oxygen enhanced MRI. And these ones are kind of neat in that they don't involve any fancy contrast agents. You're simply doing a study where the patient breathes air for a little bit and then breathes oxygen for a little bit, and looking at how the tissue responds to kind of an increase in oxygen in their the air that they breathe. And so with both of these techniques, you can kind of get an estimate of areas that are likely hypoxic within the tumor. And then the final technique that our lab has looked at are things like 18-FDG PET, which can tell us something about kind of the glucose uptake for these tumors. So here's an example in breast cancer. This is the standardized uptake value In a breast cancer patient receiving neoadjuvant therapy, before treatment starts, we see a large uptake of this radioactive tracer. And then after one cycle of neoadjuvant therapy, we see a decrease in signal. And then at the end of neoadjuvant therapy, we see no uptake at all. So this is kind of a really good marker of the function, the function of that tumor. But also you can do kind of quantitative analysis to get estimates of things like the metabolic rate of glucose within this tumor, or maybe the extravasation of glucose from the blood vessels to the tissue, and then glucose uptake by the cells. So this is, if you're going to develop a model that incorporates something about glucose, this would be a good potential imaging measurement to use. For today's talk, I'm really going to focus on using diffusion-weighted MRI and DC MRI to provide the estimates of tumor cellularity and vascular properties.
Hey, David, it's Heiko. Just a very quick question, if I may. For each of those measurements, could you comment on the uncertainty and inter-observer variability in those measurements, or how much confidence do we as modelers put to each of those measurements?
So let me. So yeah, there's a whole, there's a whole field of looking at trying to make these measurements very repeatable and reproducible. I think diffusion MRI is one of the techniques that's probably widely used across all of these measurements above here. And there are there's things like the QIN, Quantitative Imaging Network, and other networks that are developing phantoms to kind of assess how much does our estimate of the apparent diffusion coefficient vary from institute to institute, or from scanner to scanner, or from different analysis techniques. Yeah, so I could point you to some papers that would go into more detail on how that is affected, but it does come down to how you're analyzing the data. I think some out-of-the-box approaches like that are provided on scanners sometimes don't take in all the assumptions that you need to kind of make sure that these are repeatable estimates. There are kind of similar issues with DC MRI. It depends on, there are certain things you can do to make these approaches more repeatable, such as using a manual injector of the contrast agent. So you're getting a repeatable injection of that contrast agent from animal to animal or patient to patient. And depending on where you work, sometimes you are using that kind of approach, or sometimes you have issues with that. So there could be variability in that response as well. For these other techniques, for like MR elastography, tissue oxygenation approaches, and glucose metabolism, I probably do not know the literature well enough on kind of the repeatability and reproducibility of those specific techniques. I participated in stuff for the DC and diffusion weighted stuff before, so I'm a little bit more familiar with that. But the other techniques, you'd have to dive into the actual imaging method and analysis method that the lab is using, and then compare that to what has kind of been reported in the field as kind of the gold standard, the most reproducible way to do that.
Yeah, that's fair enough. Thank you so much.
Okay. So the last time I gave this talk was right before Halloween, and I had this slide in it. And since we're just a couple of days away from Valentine's Day, I thought it would still fit with this talk. So MRI is also a great tool for kind of quantifying the properties of chocolate sweets. When I was preparing my last talk, I found this really neat paper that imaged 24 different types of candies. And they looked at, different MRI properties such as T1 and T2 of, chocolate, nuts, nougat, all those kind of things. And this was kind of a fun activity on our Slack channel at work of trying to figure out what these different chocolate bars or candies were. I think we figured out pretty much all of them, but some of them, since this was a study performed in the UK, we weren't entirely familiar with all of them. Okay, so we have kind of provided a background on radiation therapy. We've talked about a little bit of introduction to the imaging modeling used. Now we're going to get into actually using this imaging data to inform these models of tumor growth and response. So first, I wanted to kind of talk about what type of data do we actually have available for these clinical and preclinical studies. For the clinical studies, patients typically receive a baseline image. So this is acquired after surgery and before radiation therapy. They then receive radiation therapy 2 grays per weekday for 6 weeks. And they also receive chemotherapy at the same time. They're then imaged at 1 month follow-up, 3 month follow-up, and 5 month follow-up. And then they also receive chemotherapy in kind of cycles following that first round of radiation therapy. So one of the things you might say is, you know, David, if you wanted to adapt or optimize radiation therapy, this is not the type of data that you'd want to have. We don't have any imaging data that's acquired during radiation therapy itself. And that's right. You know, that's kind of one of the main concerns that we have with the clinical data set right now is that we just have images before and after radiation therapy. So to kind of test out, you know, what type of imaging data we might want to have for these studies, we developed a preclinical study that tried to mimic the clinical study as much as possible. So we had this baseline image, follow-up images. The animals received radiation therapy for 2 weeks on every weekday. And then we also collected a series of images during radiation therapy themselves. So for this particular study, we aimed to collect 3 MRIs per week during radiation therapy to kind of look at the response at the beginning of the week versus at the end of the week, and seeing what kind of information we can get out of these measurements. So for both of these clinical and preclinical studies, we have very similar imaging types. We have structural and anatomical images. So these are the T1 weighted pre and post contrast images. This simply tells us kind of the extent of that clinical disease. We also have T2 weighted FLAIR, which tells us something about the invasive or subclinical disease. We also use both of these imaging techniques to provide segmentation of the simulation domain and different tissues within there, like white matter, gray matter, cerebral spinal fluid. And then we also have functional and quantitative techniques. So like the fusion weight MRI, which I talked about just a couple slides ago. This provides us something, it tells us something about tumor cellularity. And then we also have dynamic contrast enhanced MRI, which we do not have for the clinical data set, or at least for this particular data set. But we do have this for the preclinical level. So we have estimates of cellularity, blood volume, and then the extent of disease in these images. For both of these projects, we're going to follow a very similar template for building our families of models and kind of evaluating the accuracy of these models. The first thing we do is identify model candidates. So a couple slides ago, we saw, you know, that there are these 5 R's of radiobiology, these hallmarks of cancer. There's just a plethora of things that we can actually throw into models to, you know, maybe describe tumor growth. And one thing that our lab kind of leans on is, instead of maybe advocating for a single model right off the bat, we're actually going to build a whole family of models that incorporate different elements of kind of maybe response to radiation therapy, or how radiation therapy is going to be influenced by other imaging data types. And we're going to let model selection kind of play the role of deciding which model is kind of the most appropriate for this data set. Once we have this family of models, we're going to calibrate the tumor-specific properties, so things like diffusion coefficients, proliferation rates, response to radiation therapy, using a Levenberg-Marquardt method for nonlinear least squares. And for like the clinical study, that might be calibrating to the baseline and 1 month image, or for the preclinical, calibrating to the baseline and all the images collected during radiation therapy. And then finally, once we have these calibrated parameters, we are going to predict response at the remaining imaging visits. So for the clinical, that might be calibrate to baseline in 1 month and then predict 3 month and 5 month response. And for the preclinical, it might be calibrating to part of the imaging data and predicting a response at the follow-up MRIs. So the first project I'm going to talk about is the preclinical project. So for this example, we have rats that were inoculated with U87 gliomas intracranially. They were then treated with either 2 gray per weekday or 4 gray per weekday. And we're going to look at 2 different calibration scenarios. The first one is calibrating this model to the entire time course to look at how well does the data actually describe the entire time course. And then the second scenario is going to calibrate to half of the imaging data. So maybe using these first 4 imaging time points to calibrate the data and then predict what is happening at these final 4 imaging time points. The model that we use to describe tumor growth and angiogenesis is this 2-species model. The first equation describes the change in tumor cell number in space and time as a function of the number of cells diffusing, so moving between voxels, plus the number of cells proliferating within an individual voxel. And then we have an equation for the tumor vasculature. And we assume that the vasculature is going to influence tumor cell distribution because it's going to take up space. And then we're also going to assume that it's going to influence tumor cell proliferation. So the amount of vasculature is going to affect the number of cells that can be supported in an individual voxel. We're going to use diffusion weight MRI to provide estimates of this top row, and then DC MRI provides estimates of this bottom row in 3D. So here's what the PDEs for these models look like. So all of these simulations I'm going to show are going to be run in 3D using the finite difference method. I'm going to tend to show you just the central slice of the tumor, but I just kind of wanted to point that out, that these are actually 3D implementations of these models. So this first equation is our tumor cell volume fraction model. So we have a volume fraction for the tumor cells. We have our diffusion term, which has this interaction term with the tumor vasculature. And then we have this proliferation term, which has our proliferation rate, our carrying capacity for the tumor. We also have a very similar equation for the blood volume fraction. The main difference is we have this this blood vessel depth term or regression term. And then the other thing I wanted to point out is that we have this term D in the blood volume term, which is basically a normalized distance of a voxel from the periphery of the tumor. The idea is that at the periphery, this is equal to 1. So we're going to have angiogenesis at the periphery of the tumor. We're going to have that well-vascularized core periphery. And then at the center of the tumor, it's going to be equal to 0. So at the center of the tumor, we're going to now have blood vessel death or regression. One of the things that I am kind of really interested in improving on is changing how this D term is kind of maybe coupled to other information. So maybe a biomechanics model, or maybe linked back to the pressures from tumor cells themselves. For these kind of preclinical models of tumor growth and response, this kind of symmetry applies pretty well.
Ask a question?
Yeah, sure.
Hi, this is Etienne. In your vasculator equation, so you have a, in the divergence term, so not the diffusing one, but the advection one.
Mm-hmm.
I guess this is sort of the angiogenesis term, because the vasculature is migrating toward tumor in some way.
Yeah.
But the other way around, like in the tumor equation, you have like a similar symmetrical term, like the tumor migrate toward the vasculator. What is that modeling biologically?
Yeah, so I mean, so that's something that does occur in tumors where maybe it's either migrating towards vasculature or it's maybe migrating away from regions of hypoxia. So like in glioblastoma, there's a kind of phenomena called pseudo-palisading cells, where basically these cells are kind of aligning or having kind of a directed motion away from these regions of necrosis and maybe towards regions of more nutrients or more oxygen. So we are not explicitly modeling regions of necrosis or oxygenation, but this term right here in the tumor cell equation could be modeling that in some respect. So does that help?
Yeah, thanks.
Okay. Yeah, so then the other thing I wanted to point out is that our carrying capacity for the tumor is actually going to change as a response, is going to actually change as a function of the vasculature. So areas that have a high blood volume fraction, we're going to say are going to support more tumor cells, and areas of a low blood volume fraction, we are going to say are going to support fewer tumor cells. So we have a lower carrying capacity. Okay.
David, do you account for, if this is applicable to the brain, the fact that the brain has gray and white matter?
Yeah, so the diffusion term is a function of white matter and gray matter. And there's also this diffusion, the next slide I have a little bit more about kind of some other components of this model, but we do consider white and gray matter in this model. For the animals, there's not a lot of kind of white and gray matter differences in the brain, so it kind of adds maybe some minor differences. Next slide, okay. So you might say, David, this is a really, complex model of, tumor growth and response that consists of this angiogenesis term. And so I wanted to provide just a little step back here to kind of look at how we got to this point. We first started with just a plain reaction diffusion model that had global parameters, so uniform proliferation and diffusion coefficients throughout the whole domain. And this worked pretty well, maybe predicting just changes in tumor volume, but maybe not so well at predicting what's happening within that tumor. So the next thing we looked at were kind of varying the proliferation rates voxel-wise and maybe the carrying capacity voxel-wise. And then we also have this mechanically coupled diffusion coefficient, which I didn't put on that previous slide. But basically, that diffusion coefficient is going to change in space and time as a function of both the mechanical properties of white and gray matter and kind of mass effect that's produced by that tumor. So when we include all these terms, these improved our kind of ability to predict shape and the intertumor cellularity. But one thing that's a downside of maybe having these voxel-specific proliferation rates or carrying capacities is that it's temporally static. So it's going to be the same value that you calibrated for the remainder of the simulations. And one shortcoming of this was it maybe didn't predict areas of necrosis that developed outside of that calibration stage. So one way we went to try to fix this was incorporating tumor vasculature. By incorporating the vasculature term, we can add another equation that can maybe influence this carrying capacity or maybe influence the proliferation rate. And we can now maybe be able to predict these areas of necrosis in the tumor. So we now have the model of tumor growth. Now we need to have a model for response to radiation therapy. These are just a picture of the raw data that we have for these animals. So this is just the total tumor volume for our animals. So the top row here is just the animals that receive 2 gray per day. And we see some animals have very few time points. Some have a lot of imaging data available. But we see very different response across these 4 animals. And if we look at the 4 gray cohort, we see a very similar thing where some kind of flatline very quickly and others have a rapid growth and then kind of level off or flatline. And so because of this, we saw that we really needed to develop a family of models of radiation therapy to kind of study these different, to kind of model this response. So this next slide is going to kind of walk you through the different models that we came up with. Just kind of for reference, we have this first model, just untreated growth. So if there's no radiation therapy, this is what would happen.
Hey, David, sorry to interrupt. It's Heiko. I really appreciate the different dynamics you see early on during radiation on the previous slide. That's very important. Would you be able to comment on how frequently you observe those? Are those like, do you always see like the different pattern at equal distribution, or is one much more likely than another?
Yeah, I think that is, I mean, the unfortunate thing that this is only 7 animals. I think we need to do
Okay.
more studies to kind of look at, how
Okay,
consistent
sorry,
that-
but
Yeah. So yeah, we have this model of untreated growth. And the first radiation therapy model that we included is this linear quadratic model, which is kind of one of the most standard ways to kind of model response to radiation therapy. And the way this is typically applied is that immediately after radiation therapy, we have a removal of tumor cells or removal of some tumor volume, and kind of the tumor shrinks, and then it regrows, shrinks, regrows, shrinks, regrows. The next model we have still consists of that linear quadratic term, but we also have this reduced proliferation. So it shrinks and then regrows at a slightly different rate following each fraction of radiation therapy. The idea behind this is that with each additional fraction of radiation therapy, we're assuming that there's some population of cells that are maybe kind of stuck in cell repair, and therefore not they're therefore not proliferating, or maybe are becoming senescent. And then our final model we looked at was a model of both reduced proliferation, and then instead of this linear quadratic model of death, we just introduced a kind of exponential, just a death rate due to radiation therapy that changed over time. So this is kind of a much smoother curve response to radiation therapy. On top of this, we looked at an additional 3 different ways that we can actually vary response to radiation therapy. So basically this, in all these terms above here, we have this SF term, this surviving fraction term. And we looked at different ways this surviving fraction term can vary as in space and time. So the first way we looked at was this coupling approach 1, where response to radiation therapy is going to vary with cell density. The second approach is varying response with blood volume fraction. And then the third is just assuming that it's uniform across the tumor. So now I'm going to walk through kind of our model calibration and show some results from this work. So for this approach, we're using Levenberg-Marquardt method, where basically we provide an initial distribution of our tumor cells, an initial guess of our model parameters. We use our finite difference method to simulate the model at different imaging time points. We evaluate the model at those time points and compare that back to the measurement. Using the Levenberg-Marquardt method, this provides us guidance on how to update those model parameters to decrease the error. And we repeat this until our stopping criteria are met. Either the optimization has reached kind of a steady state, or it has fallen below a provided tolerance. One of the next things that we're kind of looking at is maybe looking at the confidence in our individual model predictions and the developing of these ensemble average models. So once we have our calibrated model parameters, we then calculate parameter confidence intervals for each model and each animal. We then sample these confidence intervals for these model parameters and evaluate the forward model with each set of sample parameters. So for each animal and each model, we're going to have a set of, say, 100 model evaluations that are sampling these parameter confidence intervals. Once we have performed all of this, we then perform model selection using the Akeki information criterion. And we're going to select 2 different types of models. The first one is just selecting the model with the lowest Akeki information criterion. So this is the model that best balances model complexity and model accuracy. And then we're also going to construct an ensemble averaged or weighted model. So here's an example of model weights for the different models that we have. And I'll show actual weights on a later slide. So the first question was, how well does it fit the entire time course? So looking at the global level, just the tumor size and shape. So we have percent error in tumor volume for all 7 animals for the selected model, which is orange, and the ensemble average, which is blue. We see that there are significant differences between the selected and the ensemble average for all but 1 animal, animal 4, and that the kind of median error is between ±5 for most of these animals, with the exception of animal 6. But I would say, qualitatively, the ensemble average is not that different from the selected model, and that there are some cases where it actually has reduced error compared to the selected model. The dice correlation coefficient tells us something about the level of overlap between the predicted and estimated tumor volumes. We'd like this to be as close to 1 as possible. So we have a cluster of animals that are really close to 0.9, and then the remaining are between 0.7 and 0.8. So we're doing pretty good at predicting, describing that tumor shape. When you look at kind of how well it does at the voxel level, so looking at prediction of tumor volume fraction or blood volume fraction, on our left, we have tumor volume fraction. We have our Pearson correlation coefficient. We see that it's generally 0.8 or higher, so we're having good correlation. And then our concordance correlation, which tells us the level of agreement at the voxel level, is generally 0.6 or higher. So we have some that are doing very well, and then some that are just doing okay. For the blood volume fraction model, we see a very similar story where the Pearson correlation coefficient is generally above 0.7. There are some model evaluations that are doing much worse. And then the concordance correlation coefficient is generally above 0.6, but there are some, again, that are doing a little worse here. One of the things that's kind of interesting to look at is actually the model weights for all of the models and all the animals to see kind of if there's any ones that just seem to continue to be selected. So we had 18 models in total. The first two kind of main groups were looking at whether or not we should fit for a local proliferation rate, or we should just say the proliferation rate is uniform across the tumor. The next level is looking at the 3 different radiation therapy models. So that is the linear quadratic model plus reduced proliferation plus cell death. And then finally, we have the 3 different ways to vary radiation therapy within the tumor. The main thing to notice here is that basically models 7 through 9 had the highest weights in this approach. And these are the models that had both the reduced proliferation rate and reduced death rate. So the next question is, how well does this actually predict response at the global level? For this,
Hey, David, I'm sorry.
Yeah.
If you could go back a slide, can you explain a little bit or go into more detail as to how you found the weights for each of the models?
Yeah. So there's a formula for taking the Keiki information criterion, and basically, I think it is maybe the difference between the minimum AIC and the individual AIC for a given model divided by the summation of the AIC. So there's basically for each of these models and animals, we calculated an AIC, which is the Keiki information criterion. And that considers things like the residual in the model fit and then the model complexity, so the number of model parameters. And from that, there is a conversion from that AIC to a model weight based off of kind of the minimum AIC that you see in that image.
Okay, thanks. And then is there a reason why the weights are cut off at 0.5? Like, is it possible that any model could be weighted higher than that, or is it just based on the formula that you're using?
It's based off of the formula. So yeah, it could potentially, there could be one that is very close to 1. This is just scaled to basically the maximum. I think this one right here is maybe around 0.45. But for this, to help see kind of have some contrast in this image, I lowered the bar down to 0.5.
Okay.
Okay, so
David,
how well
David, do these weights have to add up to 1?
The weights have to add up to 1, yes.
So if you go across a line, then the sum of all these beautiful colored squares should add up to 1. Is that right?
That's right.
Okay, thanks.
So how well does it predict response at the global level? So for this, we only had 5 animals that had significant, substantial time points for us to actually calibrate half of the data and predict the remaining half. And so we look at the prediction of kind of the second half of the data. It does really well at predicting this. There's some animals that have, kind of higher error, around 12%. And we notice that there are some of these where we actually don't see the ensemble average at all for 1 and 6. And that's just basically where this this box is cropped at. The ensemble average was way outside of this error range. And so to kind of see the results for the selected model, I did not present that here. So some of these ensemble averages do really poorly. And our dice correlation coefficient does similarly well, where most of them between 0.6 and 0.8. So we have a good agreement in our prediction of that tumor shape. At the voxel level, we kind of see a similar story. The Pearson correlation coefficient for the tumor volume fraction is greater than 0.6 for most of the animals, and most of them are kind of hovering around this 0.7, 0.8 range. Our concordance correlation coefficient, we see that there are some of the selected models that have very low agreement at the voxel level, but then the ensemble average models are actually doing pretty decent at agreement at that level. We kind of see a similar story for the blood volume fraction, where the PCCs, the Pearson correlation coefficients, are greater than 0.6, maybe averaging around 0.7 here. And then the concordance correlation coefficients are just lower for those models as well. So here are kind of the results for all the animals for the prediction phase. So we have animal 1. This is the measured tumor volume fraction, the selected model, the ensemble average, and then this is our blood volume fraction, measured, selected model, and ensemble average. The first thing to look at is visually, there's not a lot of difference between the selected and ensemble average model within this particular slice. I think for some of the ensemble averages, it predicts more spread outside of the slice. So it maybe is covering more slices. But there are slight differences if you squint, between the ensemble and selected model, maybe just kind of in areas of necrosis. For animal 3, we see that the ensemble average model is predicting a kind of lower cell density periphery on this tumor. For animals 5, 6, and 7, we kind of see a similar story. Generally, the ensemble average looks very similar visually to the animals, so this is not really a great way to kind of judge the differences between the model. They look visually similar in the same slice. But one thing that's interesting is for animal 6 here, where we see that even with, I think there was maybe 1 or 2 animal, this is the one I believe that had a very high weight of maybe like 0.5 for the selected model. But even for the selected model, there's enough weight from the remaining ensemble average that it has a very high cell density in these other areas of the tumor. The blood volume fraction model, you can see that there's the ensemble and the selected model perform very similar. And the results definitely depend from animal to animal, how well it does at actually predicting what's happening. So some future directions for this part of the work. This is one of the last experiments that I ran. And I'm kind of anxious to get back in the lab and run it on a larger cohort of animals to kind of see if there are different variations in response to radiation therapy, especially early on, and then trying to track down where that's coming from, like Heiko had mentioned earlier. Also, kind of further model development and validation. The experimental design itself, I think, is kind of challenging on the animals. So I would also like to go back and kind of improve the experimental design of how we're giving radiation therapy and imaging them. And then finally, I might look into other model weighting schemes to kind of develop this ensemble average. Because you saw for that one animal that the ones that had really poor predictions definitely influenced the ensemble weighted average and overestimated that tumor shape at the final time point. Okay, so I'm gonna get a sip of drink here. We covered the preclinical stuff. Now I'm gonna talk about my clinical work. I'd like to say at this point, please do not tweet or share the remaining portion of the help with this project, and of course our collaborators at MD Anderson, and then funding that supported these 2 projects. If anyone has any questions, I'm glad to take them.
Hello, David. This is Daniel. Thank you for this talk. It was really great. So one question is, well, I was thinking about it really with the clinical on the clinical side, but also you could say non-clinical. Have you looked at comparing your model fits to just a static model? So if you just propagate the image forward, how much does your model really improve?
Yeah. So I definitely have done that for the preclinical model. That's a question we've gotten in reviews before. And so for the preclinical model for responsive radiation therapy, there is, I want to say, I think there was a slide that I think was my radiation oncology paper from 2020. I think I have a figure that talks about, that shows if we just assume the tumor doesn't change from that calibration, how much error do we get in our prediction? So there is significant difference for the preclinical model. There is change there is change in the tumor volume from visit to visit with the clinical, but it's not something I have kind of off the top of my head of how much it's actually changing. But I'm in the process of making figures to look at that, because I recently got a question about that as well. So.
Okay, thank you. That's great. And then my second question, which is actually a more important question, I think, is correct me if I'm wrong, I understood what you want to do is to predict how the tumor is going to look during radiation, right? And how it's going to change. So you can adapt your radiation, your extent of radiation. Is that right? Okay.
Yeah.
But then you're calibrating with the data, with the preclinical during radiation, but then you're predicting all the way out. I'm just wondering if that was really necessary, if maybe it's more important to get the the tail end of the radiotherapy.
Yeah.
And then again, to see if just propagating from just like a static model how much benefit does it have? Those were some of my thoughts and questions.
Those are both good questions there. Yeah, I think one of the challenges with this, kind of analyzing and trying to summarize what's going on with this preclinical study is picking out what are the relevant time points to kind of evaluate that prediction.
Yeah.
And so, I think my initial thought was like, oh, we're going to predict at the end of week 1, end of week 2 of radiation therapy, and then long-term response. But since we have such a heterogeneous, experimental study here, I ended up just dividing it into just predicting half of the data, calibrating with half, predicting the remaining half, so that it's kind of a consistent formula between all of them. But that is, I think if I was just to look, for some of these animals, if I was just to look, I think for like the preclinical work, like this one has, 11 days that we're predicting. If we're just predicting, maybe the following week, it does pretty decent, and even just maybe the error would be lower in our kind of summary plots before. But I think that's just one of the challenges. We're trying to figure out the best way to kind of summarize this data with such few animals, basically.
Right. So. Right. And then just one really last question, just real quick. I was thinking about it in the beginning. Have you considered looking at 2 baseline images rather than just the one?
For the preclinical or the clinical?
The preclinical. Clinical is probably going to be very difficult.
Yeah.
Well, you don't have it, so.
So for some of the preclinical animals, so my typical framework was for this study was to image on Friday, and then they would start radiation therapy on Monday. So I would actually collect another baseline image on Monday before they did radiation therapy. So we do have a few baseline images for that. Some of these animals had such large tumors that I actually had to start radiation therapy on Friday. Friday, Saturday, Sunday, Monday, Tuesday, just to keep them in the study.
Right.
But yeah, that was the original goal, was to have those 2 baseline images.
Okay. All right. Thank you.
No problem.
Hey, David, that was really nice. Thank you so much. I have a couple of questions for you. When you evaluate your prediction accuracy, you introduced your dice coefficient and the concordance index, and you compared always against the actual volume. What is a measurement? What is a prediction performance that would be actionable for a clinician?
That is a good question for me to ask my collaborator at some point. I think you asked me that before too, and I have still not, followed up on that yet. It's on my notebook somewhere.
Yeah. So the thing is, if you were and even if you know what you're targeting for, right, what prediction accuracy, then I would like to come back to the thing of the measurement error. If you delineate the volume on an MRI, and how much variation, if you give the same clinician the same image 5 days in a row, where you get 5 different answers, and then how much of a deformation or a change in the tumor volume would you need to predict such that one would consider replanning?
Yeah. So we've looked at that in our breast cancer study. Angela Jarrett has been looking at, kind of uncertainty in our ROIs and how that error propagates through the calibration process, through the prediction process. And that's something I need to do with the same level with my preclinical and clinical work too, to kind of see if that tracks with what we're seeing in the breast cancer study. Yeah, so those are definitely, I feel like those are the questions we need to answer, before we are going through and actually, we need to add more confidence bounds to our predictions, basically, that are including information about the uncertainty in our measurements, both in the regions of interest and the ADC. And luckily, there are people at our institute that are really good at kind of taking that account in the predictions. So hopefully we're going to leverage their experience with that. So.
Yeah, but hey, congratulations on getting that work done with Dr. Chung. Yeah. Caroline, right? Yeah, we really look forward to seeing the data that comes out of that. That will be a goldmine for a lot of these kind of studies.
It's exciting. It's a good collaboration.
All right, any remaining questions for David? Sandy put his video on, that's a good sign.
I have one, David. So thanks for giving us the walkthrough here. It was nice to see what you've been up to these last few years. I guess one of the things that intrigues me, you didn't really touch on it that much, but harken back to my angiogenesis days, I can't help but wonder what your baseline angiogenic model or network looks like as these tumors grow into that. And in relation to that, what do you think about the actual non-tumor tissue here as being a player? Because obviously it's not. And that also relates to the vasculature too, right?
Yeah. Yeah, so for the angiogenesis models, we don't necessarily have, we don't resolve fine vessels within this framework. For some of for the non-U87 models, they typically have these very well vascularized periphery and a low vascularized core. For this particular animal, it's kind of very heterogeneous. We've definitely thought about ways to, yeah, one of the things that's tricky is kind of how is that vasculature being co-opted from the normal vasculature than healthy brain. And I think that's just so work that we need to do. We have some kind of cell scale model work led by Caleb Phillips that I think hopefully we might be able to kind of link to this model at some point to kind of better describe the interaction of those two parts of the domain. But right now we don't necessarily have a good answer for. I don't think we have enough information to kind of inform how it's moving from that normal to healthy vasculature, normal to diseased vasculature, if that makes sense.
Sure. No, I mean, it's a tough problem to wrestle with. But I guess that, you know, the brain is being vascularized because the animal's functioning and it's working, right? So that would imply that the normal is fairly well vascularized tissue that it's invading into, right? So I guess that, you know, a simple way to think about this is that there's some baseline normal vasculature throughout, and you're invading into that. And so when you're growing these tumors, and do you have some baseline normal throughout, or is it just angiogenesis, new, you know, neovasculature that's occurring?
So when we were growing these in silico or
in silico, yes.
No, for this we are not considering normal blood volume or normal healthy appearing blood vasculature in the model. So that would be definitely, we could probably include a kind of average value for white matter, gray matter. There are literature values for that, and evolve the model forward that way.
Okay, thanks.
I have a question about the like sort of model model generation aspect of this.
Mm-hmm.
So you had like some models that, if I understand correctly, you calibrated from the mouse data you're working with. But I'm wondering if, like, going forward in the clinical collaboration, like, one can imagine that there might come a patient that doesn't really match those models, and then you would have to potentially come up with a new one, or maybe, you know, like you're saying, kind of combine them in a smart way. I'm wondering if you have any sort of plans, future plans in the pipeline for how to recognize when a patient turns up that isn't really being fit well by the models, and like how to figure out which one to include next, that sort of thing.
On like what additional terms to include next for a given model, basically. Yeah.
Yeah, or like how you know when to make that decision, like if you've sort of thought about like it as a pre-plan for that eventuality.
Cool. So I think for most of that, it kind of happens as we're going, you know, looking at, you know, kind of with our model generation, you know, we first were looking at predicting tumor volume, and then trying to predict tumor shape, and then tumor heterogeneity within there. And so for those things, there are kind of key phenomena that we can maybe look at to improve the shape or what's happening within the tumor. So there's kind of a, there are some frameworks to kind of pick from that. And so like for the clinical project, things that you might want to include would be things like necrosis, maybe edema or hypoxia. But for kind of our lab, we tend to want to make sure we have some imaging data to back that up, to kind of initialize those other species or other terms, rather than just adding them off the bat. So that's another thing that we consider. If we don't have the data, we might not add that model in there. But if we have available imaging data, so like for the clinical project, we're going to start to get vasculature information. And so that would be something we can now throw that in there to try to improve our predictions. But right now we don't have that for most of our patients.
Thanks.
Okay, if there's any more questions, please feel free to reach David by email or on Twitter or by snail mail. David, thank you so much for a very excellent seminar. Thank you all for attending, and enjoy the rest of your week.